own way to benefit and to target a particular specific DNA sequences. And the system is so fascinating and it's so simple and versatile that most of the lab has shifted towards using CRISPR-Cas system altogether. So, wasting time, let's start. And since last few days I'm having this problem of internet, and that is specifically you see some lag when there is this video is on. So what I will do, I will switch off the video, and whenever is required, I will switch on and answer the questions. Okay. So let's start this whole topic of CRISPR-Cas system. So basically, we will go from the first, like how it was discovered. And for CRISPR-Cas system, the story starts with this paper. So this is the first paper in year 1987 when a Japanese group, they were studying a gene, namely IAP gene, that stands for alkaline phosphatase isozyme gene, and they were studying this in E. coli bacteria. And what they observed in this paper, that they observed that the sequencing, they actually sequencing, and the sequencing resulted a piece of E. coli chromosome, what you see here, next to the IAP gene. For example, this is the stop codon of IAP gene, and next to that they identified this locus with repeated sequences. So imagine we are actually studying basically the repeated sequence. Like in zinc finger proteins also we saw repeated proteins, and then he last chapter also talon we saw repeated sequences, and in this case also the first observation was repeated sequences that were, so this is the repeated sequences as you can see here, and these are the direct repeats of 29 base pair what they spurred. And after that they saw that these were interspaced by the variable sequences. What you see here are the variable sequences because they are not matching to each other, and these variable sequences are very conserved in size. For example, they always saw that 32 33 base pair are present, and if you see compared to the direct repeat where all the nucleotide sequences are almost similar as you can see here, so these are the direct repeats, and what these are okay. So this was the first observation, and that means you have a direct repeat of 29 base pair and you have a variable region of 32 to 33 base pair that was reported for the first time by Japanese group in 1987. Now basically the authors in this paper at the end in the discussion section mentions this especially, that an unusual structure was found in the 3' end flanking region of IAP gene. Five homologous sequences of 29 nucleotides were arranged as direct repeats with 32 nucleotides as spacing nucleotide. The first sequence was included in the putative transcriptional termination site. So this is the first putative sequence in the translation termination site, and this had less homology than the other. So that means all these sequences were highly conserved, whereas this one was less conserved compared to other. Now, interestingly, they made an observation that a diet symmetry of 14 nucleotide pairs was also found in the middle of the sequences, but no homology was found between the. So what do they understand by this? The diet symmetry of 14 nucleotide base pairs. So when you see this conserved consensus sequences, so you have 29 base pair direct repeat. Out of the 29 base pair, exactly 14 base pair is highly, diet containing the diet symmetry. So something like this structure. So basically what they are saying here is So far, no sequence homologous to these has been found elsewhere in carrots, and biological significance of these sequence is not known. So this was the first paper, and they checked it all through. So they were not able to conclude that why these sequences are present. Okay, so after that, after the sequencing were identified so basically this is the first, and after that, like, many things changed in the last 25 years. There was so basically the next 25 years or so, with the advance of genomic sequencing, we people started to have a lot and lot more sequences of archaeal and bacterial genomes, and people were identifying these kind of repeats in lot of other places also. Okay, so from as sequencing improved, actually they resulted in lot of sequences, and you know that there is a something called database. People actually deposit their sequences in the database, and from there they started to match that what exactly these sequences actually behave. So after this, lot of these sequences were identified independently many times by independent lab groups, and giving them different kind of names. For example, if one group found that group named it DVR for direct variable repeats. Then you have TREP for tandem repeats. You have LTR for long tandemly repeated repetitive sequences, and list goes on. So basically what you see here are your spaces that are highly conserved in size, and that these spacers are variable regions. So that we have seen 32 to 33 base pair long, and these are highly variable sequences, right? So apart from that, you have something called repeats. So when you see this, these are the repeats. So if you see this, repeats are highly conserved sequences, and they are around 29 base pairs. So within this 29 base pair, you have 14 base pair which forms dyad symmetry, and mostly people actually found that this has something called hairpin loop. They will form. So we will see this later. So, so independently, many groups actually found all this naming. They provided it, and all of the repeats were found. And basically, the list actually were confined, and only in 2002 year the name CRISPR. So CRISPR already also. Although the name is written like this, but in pronunciation people call it as a CRISPR system. And this word was coined and unified in the field, and this CRISPR stands for clustered, regularly interspaced, short palindromic repeats. So this was the yes.
Sorry.
Okay. So this was the first name it was coined, and in the field this was actually approved. So in this article, basically in 2002, Janssen et al. Published this, and they identified some special genes, and these genes are associated with DNA repeats in prokaryotes. Okay, so let me start the video because somehow. So what you have here is the one publication where for the first time they also say that apart from repeats and apart from variable sequences, you have something called other genes which are also present. So what we see here is your specifically and this kind of this kind of array. So what you have here, the described was the existence of set of genes that you see here, the Cas genes, that are constantly associated with CRISPR loci. So what you see here is the CRISPR loci, and as I mentioned here, these are the different spacers. You can see here in different colors, and apart from that, you have repeats which are in white color. These are the repeats, and they form Diet symmetrical repeats and short palindromic repeats are possible to prepare here. And apart from these two sequences, a set of genes, namely the Cas genes, constantly associated with the CRISPR loci, and these were named as the Cas genes for CRISPR associated genes. So they also made an observation from this paper that the Cas3 has structural similarity to helicases, and Cas4 gene showed motifs of REC-B exonucleases, suggesting that if you remember helicases and REC-B, so these are again the repair enzymes, right? So all these enzymes were actually shown to be made by Cas3 or Cas4, and basically they suggest that it is involved in some kind of DNA metabolism or gene expression kind of system. So these were the first observation in starting from 1987 to 2002. Now slowly people actually started identifying like what are the specific spacer sequences that are important. For example, it was only in 2005 that the three articles were published in the same year, making the same discovery that the spacer sequences between the repeats, some of them are actually matching foreign DNA and specifically bacteriophage DNA. So in this paper from Francisco Mojito, they found that many bacterial and archaeal species of CRISPR array were present, and they also made an observation that bacterial and archaea carrying this CRISPR spacers that were matching or that was not possible to be infected by that bacteriophage. So what in this paper, I've just taken one table. So this table from the article, when they are studied, they are these are the spaces of archaeal species and found the sequence similarity with phages of archaea. So you know that phage can infect bacteria, phages can infect archaea. So these are the specific bacteriophages that are infecting your archaea. So M10, sorry, M100 and M2. So from this, they are able to find these are the sequence alignment from the databases. So that was the observation in Francisco Mojica paper, right? So now coming to the second important paper from Porcel et al. They studied spacers from gram-negative anaerobic bacteria, namely Yersinia pestis. So what they found from this Yersinia pestis, so they also showed some of the sequences of spacer regions, and via blast they found that two-thirds of the new spacers had homologous in a region corresponding to the phages sequences. So they predicted that CRISPR locus takes up some pieces of foreign DNA, that means pieces of your phages, and and put it into the CRISPR array as a part of defense mechanism, and they speculated that CRISPR represents a some kind of memory of past infection or past genetic aggressions. So this was their observation, their proposal in their paper. So now coming to the third important discovery from the same year 2005, that Alexander Bolotin in their paper, they again study spacer sequences, Cas gene, as well as CRISPR arrays of Streptococcus strain, and found homology of various spacers with extra chromosomal elements, and mostly with the phages, and provided that hypothesis of cell immunity. That means they are proposing that these infection they are providing, some kind of immunity to the E. coli system, or the in this case it is a streptococcus strain. This is the figure from bulletin paper. So what you see here are some kind of spacer regions that has been taken from phages. So all this represent on the dot represent here. These are the portion which has been taken from the portion of the phage gene. So these are acquired from the phages. So all this paper actually suggested that some of the sequences are matching with the phages, and they also propose the not the mechanism, they also propose the theory that this should provide some kind of immunity, or this should does not allow any infection, and some kind of memory system should be there. So but in general all these three paper, for the first time they reported this, and although this was all theoretical but gave some important evidence or some clue that where the work should go on, right? So although these papers were all theoretical and bio work, and there were no experimental work to prove this kind of theory. Now, the first experimental work that was done to prove that CRISPR is kind of, immunity based, or some kind of, avoiding infection from the phages, came from the first paper published in Science in 2007. So this is some kind of landmark paper in this CRISPR group, where in the Barongo from the lab of Philip Howard published this in science paper. And what actually this paper is also kind of important. Why? Because first it is not coming from the academic background. This paper came from an industry namely Danisco. So Danisco is in Danisco. So basically, Danisco is a company which works with the milk cultures, and they are involved in the production of cheese, high amount of yogurt production. So all this basis of work they did in their company. So what is interesting here, that for an industry who is working on fermentation of cheese and yogurt production or milk culture, so for them it is very important that the bacterial culture or the strain they are using should be very pure strain, and these strain or the starting culture should not be infected by the phages. So if since they are working with the milk culture of very high amounts volumes, the volume goes in 1,000 of liters. So just a small infection can cause them a lot of cost, if cost, and if phages contamination occurs, so their whole culture will spoil, and the lot of cost will be involved in that kind of process. So they maintain a very high autoclave kind of scenario and sterile system. So what they observed in their thing, so what they use especially in selecting strain, and especially in this article they used a streptococcus thermophilus that was resistant to bacteriophage. And what they found in the CRISPR locus, for example, this is the CRISPR locus, they actually found that the CRISPR was changing by adding novel kind of repeated sequences. For example, if they started with this sequence, this is the wild type, it acquired some kind of spacer here, it acquired other spacer, it acquired these spacers or other spacers. So now coming to the experiment, what they did, I will show you. So specifically, they did this experiment where they infected the bacterial streptococcus strain with two different kind of bacteriophages. So one bacteriophage is namely 858 and another bacteriophage is namely 2972. So, and they noticed that the CRISPR system actually was capturing novel sequences that match the genome of the bacteriophage. So, for example, when it acquired um, S1 from the bacteriophage 858 and S2 from 858, it became only resistant to bacteriophage of 858. So this is wild type, it is not resistant, this is value 1. Whereas when the spacer was incorporated or acquired in the CRISPR locus, so what they have is S1 and S2 from the phages that give them the protection. So this is the protected, whereas other they are still sensitive to other bacteriophage, namely 2972 here. So in the second case also, when they acquired some sequences from, for example, S5 or S4 from the strain number 2972, as can be seen here, then they are only sensitive to this particular 2972, and they are not, resistant to 858 sequences. And there are certain sequences which are present in both phages. For example, S3 here, this is present in both of the phages. When these kind of sequences were acquired, so they become resistant to both of the phages. So these kind of the first experimental evidence that CRISPR system are an adaptive immune system for of a prokaryotic nature. So this was the first experiment that anybody proved in the system, that there are certain sequences which are acquired from the phages, and later on the same sequences are helping the bacteria in protecting themselves against that similar infection, similarly like our immune system what we have. So how the system works, let's go a little bit in detail. So what you see here, that this is the complete picture of many groups working on the CRISPR field, and this is just a gist of the complete picture, and I will go through it once. So that means next many years, the group started to look into the CRISPR system and describe their mechanism and the mode of action. So basically, when a bacteriophage injects its DNA into the cell, then a group of Cas proteins, basically here specifically 1 and 2, are involved in capturing pieces of DNA from bacteriophage and integrates it as a novel spacer in the CRISPR locus. So what you have here is the CRISPR locus, and what you see here is the first infection from the phage into the cell or bacterial cell. So what you then have, some of the Cas protein which goes and acquires some portion from the phage and integrates into the CRISPR locus. So this is the first step. So what you have here is L, means it is the leader sequence, or the starting of the sequences start from here. So basically this is the first system. Then this CRISPR actually locus is then transcribed. So the transcription is not shown here, but you can imagine if this is the whole CRISPR array, that the whole thing can be transcribed as a long RNA, a premature RNA, which are then processed in a smaller piece. And these, after processing, what you get is the guide RNA kind of system, guide RNA, or so to say CRISPR RNA. You call it both. So you get something called CRISPR RNA, which is the mature form of premature and this complete RNA, right? So this is the mature RNA. So what is the role of a mature RNA here, or mature guide RNA? This mature guide RNA actually guide a complex of Cas proteins to go and target sequences, uh, so the first step, so so to so the role of guide RNA is to complex with some kind of Cas proteins and allow it to exactly target to the sequences which is coming into the infection. And since here what you see, this yellow mark is exactly, these sequences are acquired from the phage infections. These DNA sequences are exactly matching to the incoming secondary infection. Upon secondary infection, it matches exactly, and based on the Cas systems, these guide RNA allows it to for complementarity based pairing, and it allows from the Cas system to do the nuclease activity, and this is degraded into the system. So basically what you have is two things. First is referred as a immunization. As you can see here, this is the immunization or adaptation or spacer acquisition. And then what you have is the immunity phase, where upon second infection or the second infection, the cell is protected by cleaving the DNA via Cas proteins. So here. So basically, what you have now is the RNA guided Cas protein. So RNA is basically guiding a protein to be targeted at particular sequence. So the whole process can be studied into three steps. So first is the acquisition step, where the piece of DNA, namely as a protospacers, are selected and integrated into the array. Then the second step is the expression pattern, where the premature transcripts are made from leader till the end, which are then processed further by Cas enzymes into small mature guide RNA. And finally, then you have the step called interference. So in this interference stage, guide RNA that has spacer sequences and has strong complementarity to the infective fudge brings Cas proteins to start cleavage and degrade the invader, so the cleavage actually happens, right? So is this clear? And the interference name itself actually gives that the interference means that the phage replication is not happening. That means it provides immunity. Basically, it is providing immunity to the cell. So I would like to know whether this is clear. This is really important. So please raise your hands if you all think this is clear.
Hello, ma'am. This is
Yes,
Shayanti. Ma'am, can
yes,
you
Shan.
explain the last part about the degradation of how it is binding and finally leading to the degradation? The last part after the
Okay,
binding with the protospecies.
okay. So a lot of hands are not there. Either they are listening or not listening. I don't know. So I've taken attendance, but the physical attendance is there. But really, if you want to give your attendance also, please raise your hand. If all of you are listening to me, please raise your hand. Vishwanathan, are you there? Vijay, Shubham.
Yes, ma'am. Yes, ma'am.
Okay, okay, fine. So the question is last part. I will come to this again. See, this is not the final version. So what I've said so far, that there are two phases. You can study this in three steps. First is the acquisition, where the sequences are acquired. It is put into the CRISPR array. Then these are all expressed into premature RNA. This RNA is then processed into the final pieces, which are called as guide RNA. And this guide RNA is then able to move this Cas proteins. This Cas proteins now together with guide RNA, they are coming and binding to the protospacer spaces. And for this protospacer binding, they also need one important motif called PAM motif. I will come to it later. So
Ma'am,
yes,
I have a question, ma'am.
yes, tell me.
Ma'am, for example, you have taken that when the phage infects the bacteria, then it puts its the part of its DNA into its array, special array or the array. So the point is that here, if number one is the part of the DNA of the phage which infects the bacteria. So in the next time when the same phage will attack the bacteria, how will the cell know that this is the part of I mean the array part of the second or third or fourth? So it will make the guide RNA for all those sequences, or means before making the guide RNA, it has no idea that what kind of phase it's being infected with. So it
Correct.
will make, yeah, ma'am.
So normally it has been seen that all these things, for example, blue here, number 6, 5, 4. So these are special spacers that gives the memory that these bacteria has been infected previously by these number of so to say just phages. Right, it can be infected by other. So these kind of sequences has been incorporated into the spacer array. Now when the same species or the same phages are infecting the bacteria, then it has a mechanism, it goes and scans through the whole protospacers. So after getting the similar complementarity, it will chop off the DNA. So that is the case. So before guide RNA, it doesn't have any mechanism to avoid any infection. Only after first infection, they get some. It's like same like our immunity. We don't have immunity against tuberculosis and other things. We get this antibody to our system so that we get this booster, right? So
Uh,
similarly like this, you are getting some kind of small pieces into the array system, and now when you have similar kind of attack, then it goes and checks so that it chops off the DNA during the interface step.
ma'am, sorry to interrupt you, but when it gets attacked with the same kind of phase, let's say, so it will scan it when it will form its guide RNA first. So for making its guide RNA, it has to transcribe the whole thing now. So it will
Yes,
make the guide RNA for all these DNA or DNA which it has acquired before for all kind of phases.
yes,
So
yes. It
okay.
actually transcribe the whole thing. So it actually transcribes the whole from leader till the end, and then it processed everything.
Okay,
So
so it will be
yes,
like guide RNA will be like means plasma cell for like go and check where it finds the affinity for that DNA and it will then cut it. Am I right?
yes, yes,
Okay.
yes. So I'm showing here only for the first one. So for example, if just for an example, I'm showing just the one phage which is infecting it. The DNA is taken, and then the same. So imagine you can have a green one here for guide RNA. So and then pink one here, or whatever, magenta, red, and yellow. So all this guide RNA will be produced. They will have complementarity cas enzymes together, and then it will go towards the interference. So Shanti, your question that how it actually cleaves the DNA, that you want to know, degrade the DNA, I will come to it later, right? So there are a lot of things we want to study in CRISPR system. So one important thing is the cleavage part, and cleavage will detail in our subsequent slide. So basically, so this part is important as in general, right? So this is basically what is giving them. So first is immunization step. So you are getting some kind of acquisition, and then once you have the guide RNA expression, or once you have acquired the sequences, then in the second stage of infection, you will get an immunity, or the bacteria will get an immunity. So this is what the work of a lot of people, and this work has been actually defined very well in the review article, namely Marafini et al. In Nature. Okay. So now coming to the types of CRISPR system. So if you
Ma'am,
see, yes,
Tirthankar and Surbhi have raised their hands.
yes, tell me,
Hello, ma'am. Am I audible?
yes.
I'm sorry to interrupt you, but I have to ask this question. Are the guide RNAs all constitutively expressed? And whenever they basically scan the entire cell volume all the time, and then when they find something complementary, the chopping happens.
Yes, yes, they scan all the time, and the chopping happens as soon as I will come to one system, and I will explain it in detail. So now I've just explained the gist of it. Like in Talon, I explained first 10 slides just for like giving you the relationship between virulent genes and our genes. So here I've just given a gist of CRISPR. Like this is how it happens. So basically, it is constitutively expressed, and then it scans all the time.
Similar to how the uh, the antibodies scan the surface of the T cells scan the surface of the human cells. Similar to that, conceptually,
You
that is.
You can imagine that, but I'm not very, 100% sure about T cells, how it works. I remember somehow like MHC, but recently I've not read anything on T cells or B cells. But you can imagine something like adaptive immunity, that once you have some kind of immunity, like some kind of antigen in it, so as soon as you have complementarity pathogenicity, in this case it is complementarity DNA, then it will be, targeting that exact DNA.
Okay, some sort of primordial antivirus system.
Yes, yes,
Thank you, ma'am.
yes. Next question.
Hello, ma'am.
Yes, tell me.
Ma'am, am I audible?
Yes, Surbi, you are audible.
Yes, ma'am. So, how does this CRISPR mechanism is distinguishing between the self RNA, like self DNA, and what is coming from the foreign particle? Like, it can integrate any sequence within these species or some particular sequences only.
That is very important question, Surbi, and I will come to it. So basically, every system has some kind of distinction. So here, the system what we will study in detail will be something like a type 2, which has been, drastically used by everybody, like type 2 streptococcus pyogenesis system, and we will study in that. So basically, there is a small sequence which distinguish between the foreign DNA and the self DNA. So like in a restriction enzyme, you have a distinction, right? Similarly, here you have a
Yes,
PAM
ma'am. The
sequences.
methylation.
Yes, you have a PAM sequences named as protospacer adjacent motif. Okay, so this PAM sequences, especially in case of type 2 CRISPR system, type 2A CRISPR system, this is especially important, that distinguishes between the self and the foreign DNA. Is that fine? We can go ahead.
Okay, ma'am. Okay,
Yeah.
ma'am.
All right. So what you see here is the diversity of CRISPR-Cas system. And now you can see that these CRISPR comes into different size and different shapes actually. They have been classified into the two classes. Basically, this is class 1 and this is class 2. And the class 1 system, if you see, they have an effector complex of Cas protein that consists of many different proteins that you see here. For example, in first type 1, you have, you have Cas7, Cas5, these HD, and these are all different Cas system. Now in the type 3 also, they have different kind of Cas system. Now coming to class 2 system is different because they have just one protein that is able to carry out the whole immunity interference step. For example, type 2 system, which we will study in detail, that has only one protein, namely Cas9 protein, and that is sufficient to carry out the whole interference step, that means the cleavage step. Okay, apart from that, they have a common Cas protein in all the cases, for example Cas1 and 2, which is important in the adaptation. So basically here, and to tell you more, that there are a lot of groups who are working on CRISPR Cas system, because entirely it is not clear that how the system works. One of the system what people use is here, the type 2A system, and we will also study that in the subsequent slides. So basically what you see in this slide are the classification that has been classified based on the number of Cas proteins which are important for the interference step, that means the effector molecule that are involved in the interference step, and Cas2 and Cas1 is important. Now, if you look further, Cas1 and Cas2 can further be subdivided into many types and many subtypes. So this is really daunting task to know that what are the different CRISPR system. So basically, I'm not going to go in detail because I've not studied all of them in detail. So basically, what you have is more than 20 subtypes that has been described, and interestingly they all differ in the way they carry out the many aspects of their RNA biology, CRISPR biology, starting from the RNA processing to the interference step. And it is important to know there are a few things to know here, that there are some system, CRISPR system available that targets only DNA. As you can see here, these are the system they target DNA, but there are systems that they are able to target both DNA and RNA, as you can see in this case. And there are systems, CRISPR system, which are only able to target RNA. So basically, if you know the whole CRISPR biology of the current CRISPR system, then one can use this whole mechanism into, you know, your own benefit, like how you can target to specific RNA, or how you can target to specific target site. So basically with this 10 to 15 slides, I'm able to convey that how the field started from the first article where they were studying the alkaline phosphatase gene, then the origin of spacer they were able to find with the help of bioinformatic assay, and they actually found that this is extrachromosomal part which is coming from the invader, and especially a lot of groups they actually showed that this is the some kind of phages they are involved, these sequences are coming from the extrachromosomal phage system. Now, after that, we saw the complete, like how a CRISPR system works. That means we can study the system in three, distinguish it into three parts. One is starting from, acquisition state to expression state to their interference space. So then once the bacteria acquires this DNA or protospaces, then they are able to offend itself against that pathogen. That means they are resistant to the selective phages that we have seen. And the experimental system was shown by the UNESCO group, where the Barongo lab, they actually showed this beautifully, that acquisition of particular spacer or cognate spacer from the phage is able to give them the protectivity against that one. And then in the end of the slide, we have seen that we have the diversity of CRISPR-Cas system. That means it is really a lot of subtypes are present, and lot of different subtypes and other types are able to give you the diversity, that you can target either DNA or RNA with this question. So since I've taken few questions, if you have some more questions, you can ask. Otherwise, I would like to finish few more portions of type 2A streptococcus. Are there any more questions on these particular slides?
Hello, ma'am.
Yes.
Sorry, ma'am, I was unable to raise hand because I'm connected through Teams mobile.
Okay.
Ma'am, my doubt is this system, like CRISPR-Cas system, gives a sort of adaptive immunity? Like, can we say that this system gives adaptive immunity to the bacteria?
Hello.